Okay, on this second lecture dealing with the quantitative genetics, we're going to really get into the nitty-gritty, at least in terms of the terms and the terminology that's involved, or this relationship, what kind of statistics can be generated and what do they mean, right? So let me get started with that. All right, so we talked about some of the history examples in the last lecture about how you might think about terms of polygenic traits and how many loci were involved. So now let's go back and think about these key components that make up the area of quantitative genetics. So we'll talk a little bit about variance. We'll talk a little bit about heritabilities as well. Okay. All right, so what do we got here? Well, quantitative genetics, again, is not about the average. It's not about really form. Now, it does question how many genes are involved, but what it's really about is the distribution, the variance, why everybody's not exactly 5'2" or 6'1" or whatever, okay? How much of that variation is genetic, right? Now, we're thinking about it in terms of Mendelian inheritance. Remember when Fisher came along in 1918 and pulled these together, he put Mendelian ideas that, yeah, there's a bunch of lots of Mendelian stuff going on, but it's combined up in ways that give us this sort of overall pattern that's because it's polygenic, okay? Now, anytime you have polygenic traits though, right? Anytime you have something that's got a lot of genes involved, then there must be environment involved, right? So there's a combination of the environmental input, whether it's the cellular environment or the internal organismal environment or the external environment, all play a role in making a phenotypic trait, right? So what we're interested in quantitative genetics really is to try to figure out how to understand this phenotypic variance, this trait, the way the trait looks, and what component environment plays in that variation, and what component genetics plays. Now, because this is genetics, we're not overly interested in the environmental component, although if you're in breeding, then that's really an important part of it. But for us right now, we're going to concentrate on the genetic variance, okay? So we're thinking about the variation in a phenotype, so the variation in this height, these heights among these individuals here, okay? How much of that variation is due to genetics and how much is due to environment? Well, what Fisher did was pulled a simple little equation. He said, okay, I need to do some math here. So I will set these up and give them some little symbolism so I can work with it. So variation that's totally phenotypic, V sub P, is a combination of V sub E plus V sub G. All right. Now, what are we measuring here? Okay. This is the total phenotypic variance for a character. And it's a function of two things, just like we said. The genetic variance, the variance among the mean phenotypes for given genotypes, right? So we had that big distribution. So we got polygenic, we can get lots of different genotypes, and we sort of have married groups of those. What's the variance? And then the environmental variance is a member expressed among, okay, within the same genotype, how different are they? Okay, we've got a genotype 24.5, and all the individuals that happen to have that genotype might be big A, little a, big C, little e, all that. Some of them are 6 foot tall, some are 5'11". That's the variation. That's the environmental part of it. Okay. Where do we get all this? Well, a lot of times we do it with twins. Okay. Twin studies have been one of the big things that in humans that have been used for looking at this variation. We can't, we don't have lots of progeny. We don't have a situation like fruit flies or something where we can have thousands of progeny and we can measure the variation from a single set of crosses or whatever. But one that works really well for us is our twin studies. And these twin studies are particularly, we're talking about monozygotic twins, of course. These, if you raise them together, okay, the twins are raised together and you look for any differences between them, it must be environment, right? And so they use twin studies where they're raised together. They also use twin studies where the twins are monozygotic, but they've been adopted out. So they're in totally different environments. So you have the sort of the extremes of that. But either way, they're using monozygotic twins. That's not the only way we can do it. We're going to see some other methods where you can use any group, as long as you have some idea of the parental phenotype versus the offspring phenotypes. But twin studies have been very, very useful. You'll see those in the literature a lot. All right, so we say, okay, we got this phenotypic variation. Just to add in another component to it, we have the environmental variance, the genetic variance, and then there's an interaction effect for the most part, okay? So what's the interaction effect? Well, that means that the environment and the genetics don't interact the same way every place. People understand that. If you take an individual that's got a certain genotypic makeup at sea level, and you raise them to say 6,000 feet, 7,000 feet, where the oxygen tension is different, or maybe the solar radiation is different if it's a plant or whatever, even though they got the same genotype, the environment may interact differently at those two places. So that's this interaction effect. Very difficult to deal with and hard to understand. So we try to set up experiments where we can X that out in many ways, okay? One of the things that we'll do that is twin studies, okay? All right, so here's the word we want to deal with called heritability. We're going to talk about two forms of heritability. One's broad sense and one's narrow sense. Broad sense is just what it says. It's in the broad way of looking at it. It's usually illustrated either with a big H or a big H with a squared or whatever to indicate variance. But what it really is the genetic variation over all of the phenotypic variation. That's really all it's saying. It's a simple little ratio of saying how much of the variation in the phenotype, how much of variation in this whole distribution of height or whatever is genetic, okay? Totally genetic. Okay? And it's very important to understand. It's the first thing you calculate when you're trying to do one of these quantitative studies after you've tried to figure out how many loci are involved. You calculate the broad sense heritability. Okay, if you're studying a trait, which the broad sense heritability, all the genetics that you have, all the different modes of inheritance and all the other things you have is 0 or 0.001%, then you're not, everything else is environment. So why are you studying it through genetics? If it's 80% or something else, if it's a big number, then you say, well, okay, there's a lot of genetic relationship to the variance that's here. Okay, now even if you get a broad sense heritability of 0.001 doesn't mean there's not a genetic component to the trait. It's saying there's not a genetic component to whatever variation I'm seeing in the trait. Okay, so just be careful when you see that. All right, what does this assume? It assumes environment is fixed, which means that this interaction effect is essentially 0. Okay, it is well known that it could change with the environment, because when you change the environment, then you do bring in this interaction effect, this over here. Okay, and again, I want to just emphasize, and I put this in here, measures this broad sense heritability measures how much of the variation is genetic, not how much of the trait is genetic. Okay, not how much is passed on. Yeah, all right, so that's a big difference. Okay, so this comes up when, there's been so much fight about IQ, whatever IQ is supposed to be. How much of it it's nature nurture, right? How much of it nature would be genetic? How much is just genetics? That's what Galton was interested in. He thought a lot of it was mostly genetic, and therefore you could modify it by breeding and doing all sorts of other things. Other people say absolutely not. We're all, if we're given the same, the proper environment, the proper training, our genetics plays a minor role in what we would call IQ or intelligence, ability to succeed and think and do things. So there's nature nurture arguments, and there's been all kinds of controversy for, just ever since Galton. The bell curve, and some people are here, and then they always throw in racial things to it and cause all sorts of calamities about it. But what we're really talking about is there's clearly variation in IQ, right? Some people have IQ of 160, some people have IQ of 80. Is it how much of that variation is genetic? How much of it is because of other environmental inputs? All right. So what are some examples? Well, from twin studies, okay, so taking monozygotic twins, identical twins as we call them, let's look at some broad sense heritabilities. One of them is height, okay? Our height is apparently, most of it's genetic. It's not a lot of environment to it. What does that mean? That means that, okay, I have a son or daughter and I want them to be professional basketball players. So they need to be 6'8", 10, 12, whatever it is. Okay, I'm not. I'm 6 foot, or I'm 5'9", or whatever my height happens to be. Both of us in the couple are the same, close to the same height. What's the chances of getting a 6'10" child? Well, genetically, it's not likely to happen. Okay, we're going to probably not. And what it's really saying is only about 12% of the variation in height is due to environment. So I can feed that kid all I want or make them jump or do whatever I think is environmentally going to manifest itself, but certainly nutrition would be the thing. And they're not going to make it. They're not going to go from being 5'10" to 6'10". Okay, it's not going to happen. Okay. Other things that are, that will change are waist circumference, how large around the waist. That's only got a 25% broad sense heritability. About 25% of that is genetic, and so variation that you see, some people have really lots of material around their waist, others don't. 75% chance. So if I want to modify that, I can simply give people less food or more food. Makes perfect sense. We've all seen that. Finger ridge count, whether they're quirls or tints or cones, whatever you happen to have, that's highly genetically inherited influence. And other things that go along with that. And some things are high, okay? You can ignore this IQ, that's just a rough estimate thing, but acquisition of information, it's not as high as processing of it once you get it. Some behaviors, they do have heritability. That's, which some people would argue that they don't, that they're all learned, but in fact, whether you're extroverted or introverted or whether you're antisocial or whatever, those have some at least genetic component. That also means that they have a large environmental component because the genetics here is only counting for about 50%. Others, there seems to be things like autism and schizophrenia. We'll talk more about some of those later as what pathways might lead to that and why there would be a lot of genetic component to it of the variation that we see. Again, we're talking about variation, right? Other things, not so much. And then things that have heritability that you wouldn't think about, okay, is this choice of religion, those things. Now, that probably doesn't have anything to do with direct choice of religion, probably has to do with ethnicity and the background that you would have and those things, okay? So people just throw those in and say, oh, okay, how is those heritable? Well, there's variation in that and some of it's genetic. Well, because of the real background in certain groups, right? That come along from the form. So it's an interesting social discussion that you put into that. All right, so now that we've got this broad sense heritability, right? We've taken this idea that we want to take this genetic component and we want to look at it, okay? And we have some measures that there's a genetic relationship there, okay? How do we really use it? All right, there's a lot of stuff that will cause genetic variation, right? There's a lot of modes of inheritance, there's epistasis, there's all sorts of things. Well, we can take that genetic variance and we can break it down into some other subcomponents, right? So classically, so what we're doing here is we're just taking genetic variation. It's here, then we're saying, okay, that genetic variation has multiple components that could possibly be. We're going to focus on 3 of them. That's the classic ones that we see in quantitative genetics. These are additive variation, variation due to dominance, and variation due to interactions, such as epistasis, okay? So the question becomes, okay, we can now parcel these genetic variation out into additive, dominant, and interactive. What the heck does that mean, right? Well, it turns out that for us and for most breeding studies and things where you would actually go out and do sort of agricultural uses, the additive component is really one of the more important parts, okay? Because dominance is pretty straightforward. If you like the tall ones and that's the dominant trait, you just select for the tall ones. But additive is a completely different little model. So what's the difference between additive and dominant gene action? Well, dominant gene action is just what you say. You take these very simple genotypes here, okay? Big A, big A, big A, little A, little A, whatever you want to call them. Okay, I put them as B's, but or they have from this paper. And you say, okay, dominant effects is that once you get a certain genotype, in this case, B, all right, big B to the second or little b, whatever, a sub 2, you're dominant. Okay, that's it. That's all you get. That's dominance. And again, if I want whatever B2 produces, I just make sure everybody's a B2, at least has one copy of it, and I'll get that. Additive, though, is that it adds up. Each component adds up. Each allele has some effect. okay? Dominance could be 0 or partially 0, and you just simply add on. Now, with a two allele system, one locus, two alleles, this looks like incomplete dominance or something, right? You've got white ones and black ones, and you've got gray ones. But it gets more complicated. Remember, we're talking about not single locus systems. We're talking about a bunch of loci piling up into a polygenic system. You have additive there, you get a different outcome, okay? Additive variation there, and again, this is one of those classic examples of where you get all this sort of mixing going on, okay? Let's take an example here where we have just a two locus system, big A, big B, big B. This is where we usually try to understand this additive variation, all right? If we think about that, you get, if we give one dose of color to big A, it doesn't matter whether it's big A or big B, if you're the dominant form, you get a dose of color. This individual that got this genotype would get a dose from this big A, a dose from this big A, a dose from this big B there. You get 4 doses of color, right? Okay. And of course, now what we're talking about is taking the system and kind of mixing them. That's why I threw in this little palette down here. We start taking a little bit of this red and then put it together with blue. We get this, we put this, a little more of it with something with green, we get something else. You start mixing these doses, and then how much of this you pull down? Do I get 4 pull downs of this, or 1 or 2? And then I start getting all kinds of combinations, right? Well, in the genetic sense, though, what we're talking about is, okay, this one, big A gives 1, and we'll make the simplest case where little a or little b gives you 0 doses. That gets 2 doses, okay? That's a hit. That gets 2 doses. This individual, big A, little b, also gets 2 doses. So whatever color those are going to be, they get 2 doses of color, okay? So an individual, as you can see here, that's got little a, little b gets no dose of color. Everybody like that. Anybody that can get 1 allele, okay, gets 1 dose of color, right? So that could be big A, little b, it could be whatever. You get 2, you get doses. So you can see all the different doses of color, and these are just how many things. That's the additive component, right? Now you think about it in terms, okay, this is 2 loci, and you get all those different outcomes from those 2 loci, right? You get some that have 0, you get some that have 1 dose of color, 2 doses of color, 3 doses of color, 4 doses of color. Imagine having 10 loci, like we looked at. We said how many loci are responsible for yield in something like wheat or corn. We said, oh, it's 12 loci involved. Well, this just goes on forever there, right? We can get all sorts of, if we're assuming that there's some additive variation there, then we get a lot of different doses. All right, so this additive component, if that's what we're interested in, and we are interested in it because maybe we want to produce a lot of them that look like this, the 3s or 2s, okay? How do we do that? Well, the measure that's used for that is the second one of these. First, we had broad sense. That's all genetic variation of the phenotype. We're going to narrow that down to just the variation that's due to the additive component, because it turns out that that's the one we can play with, okay? That's the one that's sort of interesting as far as being able to modify and things, okay? So all that is the variation that's additive divided by the variation of the phenotype, total phenotype. Now there are modern ways of playing with that, but as far as the classic way that was proposed by Fisher and has been used for 100 and something years, the variation that's additive divided by the variation that's phenotypic. So if this was broad sense, this would be variation that's genetic, right? All the genetic parts, whether it's additive or dominant or interactive or whatever, we're simply focusing out. This heritability will range from 0 to 1, okay? And with traits with no genetic variability that is due to additive will have a heritability of 0, okay? So what are some examples in humans of narrow sense heritability? We want to narrow it down, okay? So we're not just saying how much is genetic, we're saying how much of it is additive. It'll add up. And things like body mass has a heritability around 0.4, 40% of it is heritable in an additive way. Cholesterol, high density lipoprotein, about 40%. Low density is much less. It's around 20%. A half of that is much less additive. Okay. Then there's things like sex ratios. Well, sex ratios are determined by the sex chromosomes, and there is variation. Okay, some families have 4 girls, and some families have 3 boys and 1 girl, and there is variation in that. But very little of it has to do with additive variation. Okay, you really couldn't change that. So what does that mean? And recombination rate, how fast it occurs. Well, these are variants. So let's say you wanted to take a population, and you wanted to increase the amount of high density lipoprotein. All right, this number tells you could do that. All right, it says that really all you would have to do is to mate individuals that have high density lipoprotein together, and you would take that distribution and you would move it. Okay, you would move it. That's the selective coefficient of that, and that's what we do. That's what we have done for millennia with breeding studies and breeding examples. We wanted something. We wanted a greater yield. We wanted less height. We wanted more, thicker stock or less thick stock. We would find those things, and if they had heritability, they would work. So this would tell us that if we wanted to modify high density lipoprotein, we would be able to do that to some degree. Low density lipoprotein be less likely to be able to do that. We'd be able to do some, move it, but the curve would not move near as far because there's not as much additive. So just doing breeding studies with that or whatever wouldn't be near as effective as it would be for high density. That's how it's used. Okay, so these are just some measures of that where you can and you see things that have menopause has various ages. Some people are menopausal in their late 30s. Some people wait till their 60s, and you have an additive component of that falls in. Okay, all right. Well, that gets us through some of the nomenclature, and that's really what I'm doing. Again, the reason I'm doing this is that we're going to come back in the second module, the second part of the course, and I'll be mentioning these. We're also going to see them in some papers where they go out and measure heritability of some things, and you're saying, oh, okay, I don't understand what that means. Well, now you at least can go back to the notes and understand a bit what that means. The last of these lectures on quantitative, I want to talk about measurement techniques. Just some ways, particularly in humans, that we can actually generate some of this information.